All right, good morning, everybody. I'm going to go ahead and get started here. First of all, I just want to acknowledge everyone's flexibility for having class online this morning. I apologize for mixing things up this week. I've had two very sick kids at home all week. Fortunately, not COVID or the flu, but it's been kind of a challenge to bounce back and forth and everything. So I appreciate your flexibility here. In advance of class today, you had some readings and a video, and we are going to continue with our current topic of the brain today. And we're actually going to wrap up the brain today and transition and tackle some new material starting next week. So today's agenda looks like this. We're going to review your ticket out from Wednesday. We're going to continue our coverage of the cerebrum. And then I'm going to show a short video. I had planned on showing this in lecture class, but what I'm going to do instead is provide the link to YouTube, and you can watch it independently for 5 minutes, and then we'll bring it back to the full class. And then we are going to talk about the protection, the layers that protect the brain, and then just some wrap-ups and reminders. So before we get into reviewing Wednesday's ticket out, oops, I didn't mean to do that. What questions do you have right now? Okay, well, let's go ahead and I've got people coming in the waiting room here still. Let's go ahead and review your ticket out for Wednesday. So I had a few questions for you, and overall, most of you got them, but I want to make sure that you're understanding the answers. The first question is, where do most of the cranial nerves connect to the brain? Now, we haven't really talked about the cranial nerves in too much depth, a little bit in lab, but the big message that I did try to impart when we're covering the brainstem is that 10 of the 12 cranial nerves attach to the brain via the brainstem. Now, remember, the brainstem includes the midbrain, the pons, and the medulla oblongata. The other two cranial nerves attach directly to the cerebrum, and that's the olfactory and the optic nerves. You might remember, for example, in lab with your sheep brain, we had the optic chiasma, where those two optic nerves basically come into the cerebrum and intersect. So 10 of the 12 cranial nerves connect to the brain via the brainstem. But this question was a little bit weird. I don't know what I was going through my brain when I came up with this one. But imagine you're a tiny little organism roaming around in a human brain. Another tiny little organism roaming around in the same brain asks you to where can you find the superior colliculi? And so the correct answer was this, the orange answer. It got cut off a little bit here, but I want to show you the full text. It basically says, find the diencephalon, then move inferiorly to the midbrain. The superior colliculi are on the dorsal aspect of the midbrain. So just to remind you of those structures, this is just a goofy way to offer a refresher of some of the structures of the midbrain. Here is the midbrain. So this is a posterior view of the midbrain. So we're looking at a brain from behind. And actually, not just the midbrain, but a few other things. You can see part of the cerebellum here. This has been kind of cut off so we can see the midbrain, as well as parts of the diencephalon here. Now, the superior colliculi are these two structures in green on the dorsal aspect of the midbrain. We have the inferior colliculi right here. And remember, these four structures together are also called the corpora quadrigemina. You'll hear both superior and inferior colliculi and the corpora quadrigemina used. So just as a reminder, those are these structures on the dorsal aspect of the midbrain. Just to remind you, otherwise, in this diagram, here we have the diencephalon here on top of the midbrain, and this is the right and left thalamus. We have the epithalamus back here. Then if we go back to the brainstem, we have our midbrain up here. We have our pons right here. And then this transitions to the medulla oblongata, which of course, moving inferiorly takes us down the spinal cord. The last question from Wednesday was this, which structure will you find just dorsal and inferior to the thalamus? Most of you actually got this wrong. The key word here is dorsal, and so towards the back, or we could also say caudal. Many of you picked, or half the class picked hypothalamus. Remember, hypothalamus would be more rostral to the thalamus. The epithalamus is actually what we find dorsal and inferior. So if we look at a cross section of a brain, this is a left lateral view of a brain cut down the sagittal plane. And just to refresh your memory, the three major parts of the diencephalon include the thalamus, which is most of the diencephalon. Then we have the hypothalamus here, which is kind of inferior and anterior to the thalamus. And then we have the epithalamus, which is back here, which is kind of inferior and posterior to the thalamus. So just refresh your memory about those structures of the diencephalon. All right, let's go back to this overall schematic. We've seen this a bunch of times. Remember, we have these developmental regions that develop into adult structures in the brain. We've been covering everything here up through the cerebrum, and we're going to wrap up the cerebrum today. So before I jump into that, I'll pause for a second and see what questions you have about those ticket out questions from Wednesday or anything else, and we'll go ahead and dive into the cerebrum. All right, let's do it. So we left off, just as a reminder, you have a study guide for this section of lecture as well. And just as a refresher, I'm going to post this link in the chat in Zoom, so you can grab that if that's easier for you. You've been following along, you probably have this already. So just to refresh our memory, we were talking about the sensory areas of the cerebral cortex last class. Today, we're going to transition and talk about the motor areas. So these are the cortical areas. Remember, we're talking about the cortex of the cerebrum, that gray matter on the outside, or the most superficial layer of the cerebrum. And they control the motor function throughout your body. So this is voluntary and involuntary motor control. And all of these cortices are found anterior to this central sulcus right here. So we have the primary motor cortex, what's called the premotor cortex. We have an area called the frontal eye field. And then we have this area called Broca's area, which I'll get to. That has to do with speech. We'll talk about that a little later. But first, I want to talk about the primary motor cortex and the premotor cortex. The premotor cortex, you can see in this kind of light orange here. This is the area that kind of basically helps you plan what your movements are going to be, depending on sensory information that you've received. So this is like when you're playing outfield and you see a fly ball in the air and you're trying to judge where to go in space so that you can be underneath that ball when it lands. This is the premotor cortex that's allowing you to integrate the sensory information and then plan ahead for what those voluntary movements are going to look like. And you can see here that the premotor cortex is just anterior to the primary motor cortex. Now, the primary motor cortex is what kind of immediately controls the muscle, your voluntary muscles throughout the body. And so from these neurons in the primary motor cortex extend these long axons that form these pyramidal tracts that run through the brain. We'll see what those look like actually in another slide coming up here. But basically these long tracts of myelinated axons that run through the brain down to the spinal cord. And eventually they synapse with motor neurons that then enable this precise and voluntary control of movements of the body. So this is basically what's controlling your immediate voluntary skeletal muscle movements. Now, here, what's important to note also is that these pyramidal tracts cross. I'll show you a diagram. Is that my next slide, actually? No, it's not my next. I'll show you a slide of that in a little bit. And they are contralateral. So that means that what is happening on the right side of your primary motor cortex is controlling movements on the left side of your body and vice versa. So we talked Wednesday about the sensory homunculus. Today, I want to cover the motor homunculus. And so the idea of this is that we have this precentral gyrus, this primary motor cortex, and parts of the real estate of this cortex are dedicated to voluntary muscle control of the various parts of your body. And so we can map this with something called the homunculus, where the size of the structure that you see corresponds to approximately the number of neurons that are dedicated to controlling those areas. So the takeaway message from this is that most of our primary motor cortex is dedicated to controlling the hand and fingers here, as well as our face and to some extent our tongue. We see very little real estate here dedicated to the feet, the trunk, the shoulder, even the arm, etc. And this is, if you think about where you are generally most exhibiting very, very fine motor control of your body, it's primarily our hands, which are great at fine scale manipulation, and also our faces. Now, our faces are important because we produce speech with our faces, we consume food with our faces, and also as humans, we rely very, very heavily on facial expressions. And we're very fine-tuned to reading the facial expressions of other people and manipulating our facial expressions in a way that conveys our emotion. That's why it's so hard to kind of hide your emotions when you're looking at someone face to face. Also, why it's so difficult for me to teach this class online without being able to see anyone's faces, because I can't really read what's going on for all of you in the classroom, or for that matter, in the classroom with masks on as well. It's amazing how challenging it is. And I never really realized it until those things were gone, how much I rely on that facial feedback from students to kind of see if they're getting stuff, if they're confused, if they're bored, et cetera, et cetera. And so now with being on Zoom, or sometimes with your faces covered, that's kind of all out the window. So I just kind of assume that I'm teaching perfectly and all of my jokes are really funny, which I think is a pretty safe bet. So that is the motor homunculus. And one aspect I want to compare this to the sensory homunculus. Let me see here. I don't have that slide handy. You remember with the sensory homunculus, there was a pretty fair amount of space dedicated to sensory input from the genitals. What's interesting here in the motor homunculus, we see no motor output to the genitals. And I'm using this as an example to highlight how our sensory input and our motor output don't always correspond one-to-one. For example, we have a lot of sensory input and a lot of motor output in our hands, but that kind of balance isn't true for other parts of our body, where it may be more important to receive sensations than to control via voluntary motor control. All right, let's talk about this frontal eye field. So this is this little spot just anterior to the premotor cortex. And I'm not going to get into too much details here, but basically this spot controls the eyes, the voluntary movements of the eyes, and especially when you're following a moving object. So if you're looking at your screen right now, you see my finger over here, move it over here, your eyes are following that, right? That is due to this, the neurons in the cerebral cortex of the frontal eye field here. We also have this other area called Broca's area that's just kind of inferior to the eye field. And this is a only found in the left hemisphere of the brain, generally speaking. And it is important for managing speech production and connects with also your comprehension of language. So it's interesting, there's actually different areas in the brain for understanding language and producing language. And this is why sometimes you can have you can experience different syndromes whereby you can either understand speech perfectly, but you can't produce your own speech, or you can produce your own speech, but you have a really difficult time understanding other people's speech. And that can be due to pathologies in these different areas of the brain. So Broca's area, being in the motor area generally, is responsible for producing, for speech production. Now, it's interesting, as I mentioned, this is in the left hemisphere of the cerebrum. The corresponding area in the right hemisphere actually is where we control the emotional overtones to spoken words. So you can think of Broca's area as being more about language and grammar, whereas the corresponding area in the right hemisphere, I don't know if it has a specific name, is more about how we enunciate and how we express our emotions through our words. And I'm sure you can all think of examples where you can say the exact same word with two very different intonations, and it means two very, very different things. Just try that at home, a little exercise. Just try to say the word what in as many ways as you can and express different emotions through the word what. It's a good time. Your roommates will love you for it. So that's Broca's area. I want to move now. Those are the only motor areas I'm going to talk about in any level of detail there. So again, we have the primary motor cortex, the premotor cortex, the eye field. Here, let me just go back here. The frontal eye field. Oops, sorry, wrong side. Frontal eye field, Broca's area for speech. I want to talk a little bit as well about multimodal association areas. So these are areas that are integrating the sensory and inputs and motor outputs. And we can roughly divide this into three association areas: the anterior association area you see here, primarily in the most anterior portion of the frontal cortex. We also have the posterior association, more in the occipital cortex here. And then we have the limbic association, which is deep in the brain and kind of surrounds this corpus callosum. So let me talk a little bit about what these association areas do. First, I want to talk about the anterior association area, or also known as the prefrontal cortex. This is much of the frontal lobe. And what this area does, I'm just going to kind of talk about it generally, is it receives information from other association areas, so this immediate sensory input, and integrates that with your memories of things you've done in the past, and allows you to kind of think about what you want to do and plan your motor movements. It also has close links to the limbic system, which I'll talk about shortly, but you can think of that as your kind of emotional memory. So you're basically drawing on your kind of logical memory of the past, your emotional memory of the past, and current sensory input, and you're putting all of those things together so that you can decide what to do. So as you can imagine, this is a super important area, and it determines, it's where you control your complex functions, including things like, you can see the list here: thinking, perceiving, intentionally remembering, so studying, processing abstract ideas, your reasoning, judgment, also like impulse control, your ability to control the drives that are initiated by other parts of the brain. For example, drives related to hunger or thirst or sex. Also, integrating all this information helps you with social skills and important things like humor, empathy, and your conscious. So almost kind of all of these higher level processes that really make us human in a way. And so this area is important because it's integrating all of these complex things. Now, those of you that have taken neuroscience, you'll get into much more detail with exactly how this happens. But for us, I just want you to know the general gist of the prefrontal cortex and what's going on there. With that in mind, I want to make a brief aside here and give you an example of someone whose prefrontal cortex was all but destroyed and what happened to this person because of that. But before I do, I'll pause. I do feel like I move through things pretty quickly when we're here on Zoom, and I want to see if you have questions. Also, just remind you that I will record a lecture and post it today as well. So what questions do you have for me? All right, let's talk about Phineas Gage. Some of you have probably heard this name before. He was a worker in the railroad, and basically what happened through an accident, he had an iron rod. He was in his mid-20s, and this iron rod basically blow through his skull and through his eye and up through his brain. And you can see the diagrams there of how big it is. Now, what's noteworthy here is that this rod went right through this prefrontal cortex, this area of the brain it's integrating all of this information and involved with this higher order cognitive functioning. And therefore, it really affected his personality. And what I have here is a short 5-minute video. I think what I'm going to do is just post the link here and let you view it in 5 minutes, then we'll come back as a class. I think that'll be the easiest way to do this. If I try to play it through, Zoom, it probably won't work too well. So I'm going to post this link here. It's a 5-minute video. What you can do is go ahead and watch the video, and then in 5 minutes, we will return back here and continue lecture. So I'm going to pause the recording. All right, I'm going to bring us back here. I think everyone's had enough time to watch the video. So just a good example of how, one, resilient your brain can be, and two, how injury to a certain part of the brain can affect only certain aspects of your functioning. And you saw in the video, for example, that because the injury was to this prefrontal cortex, Phineas Gage had a hard time kind of controlling his base desires and making plans and sticking with him and that kind of thing. All right, I have a few more association areas I'd like to cover in the cerebrum. And the first of those is the anterior association area. This is an area that stores short-term memory. So this is what stores basically like what's immediately happened to you within the past half minute or so. And there's three kind of working memory areas that's related to your visual memory, your auditory memory, and your executive area. And so I know one thing that I kind of do a lot is when somebody says something to me, I don't quite process it right away when they say it, and I'll kind of repeat it back to myself, and then I'll understand it. I don't know if anybody else does something like that, but that's a great example of this auditory working memory. I just heard this thing. I haven't quite figured out what it means yet, but I can recite it back to myself verbatim because it's stored in that short-term memory. Now, if more time passes, I won't be able to do that. But for this kind of short-term recall, you can do it very well. We also have here, what I alluded to before, is your limbic association areas. And I'm not going to get into this too much, but basically here you can see a sagittally cut brain on the mid-sagittal section. These limbic areas are basically these areas of the cerebrum that surround the corpus callosum and the diencephalon here. And this is an area that's involved with memory and emotions primarily. So it's where you form a lot of memories and where you process your emotions. And yeah, I'm just going to leave it at that. There's a lot more to say about that, but I just want you to know the limbic association area, part of the cerebrum surrounding the corpus callosum, as you can see here, and involved especially with processing memory and emotions. I have a little bit more on cerebral anatomy to cover here. As I mentioned earlier with your primary motor cortex, how it operates contralaterally, I want to talk about the anatomy of how that works a little bit more. And what's interesting about this is not only is much of the brain function contralateral, but also the hemispheres tend to be specialized. And so specifically, your left cerebral hemisphere is primarily involved with the grammatical aspects of language, math, logic, this kind of thing. Whereas the right cerebral hemisphere is involved with more with visual spatial skills, reading facial expressions, things like intuition and emotion, and also specifically creativity, so artistic and musical skills and things like that. And we often refer to people kind of colloquially as left-brained or right-brained. So if you're called left brain, that means you're very kind of logical and regimented, whereas somebody who's right-brained is maybe more freewheeling and creative. And that actually has some basis in how our brain functions. Now, what's important to note is this is often in about between 5 and 10% of the population or so, this is switched. And that has to do with folks that are left-handed versus right-handed as well. So we see the specialization of the brain play out in our handedness. And as you know, for almost the vast majority of the population, one hand is much more dominant and you can do a lot more with that hand than the other. Now, that's not always true. There's some folks who are ambidextrous and can do things equally well with both hands, but that's actually fairly rare. I want to talk about the anatomy of how this contralateral projection works a little bit. And for that, I'm going to get into some of the cerebral white matter. So far, we have been talking about the cerebral cortex, which remember is this layer of gray matter on the outside. And what you see here is a brain that is sliced along the frontal plane, and we're looking at it from behind. So we can see here this is an anterior view of a brain that's been sliced on the frontal plane. And so we have the cerebellum in the back here. You can see the brainstem coming up here. You have your diencephalon here. You have the lateral ventricles here. These are filled with cerebral spinal fluid. And you see a cross-section of the cerebrum here with all these sulci and gyri. And you can see the gray matter on the outside and this white matter on the inside. So the white matter is important because these are long tracts of myelinated nerves. And I'm just giving away, I have asked the question here, are these fibers going to be myelinated or unmyelinated? Well, it's white matter, so they're going to be myelinated, generally speaking. And then we have these long tracts that connect the different parts of the brain. And these are called commissures. I should be more specific. Commissures are these myelinated tracts of nerves that connect the left and the right sides of the brain. And so the most kind of prominent example of this is what's called the corpus callosum. This is the largest commissure. So it's the largest tract of these myelinated neurons that are connecting your left and right hemispheres. And this is just superior to those lateral ventricles that hold the cerebral spinal fluid. We also have what's called association fibers. These are fibers that work within the same hemisphere. So these are tracts of white matter that connect these different parts of the brain within either the left or the right hemisphere. So for example, tracts that connect parts of, I actually haven't talked about this, Wernicke's and Broca's area. So this is the area that's primarily involved with speech recognition, and this is the area primarily involved with speech production. So obviously they need to be very closely linked. And so we have these association fibers linking them in the same hemisphere. We also then have what's called projection fibers. These are running from the cortex down, most often through the brain stem and potentially down to the spinal cord. So those are the fibers that are running from the cerebrum to other parts of the brain. So just to refresh, the three fiber tracts in the white matter of the brain are the commissures, which connect the left and the right hemispheres, the association fibers, which connect areas within the same hemisphere, and then projection fibers, which run from the cerebrum down to other parts of the brain. Finally, last slide on the cerebrum is we also have, deep in the white matter, we also have some gray matter in the cerebrum. And specifically, we have a couple of different nuclei. Again, I'm not going to get into too much detail here. I just want to provide you some examples and remind you that there is gray matter deep to the white matter. So again, we have that gray matter of this cortex on the outside, white matter deep to that. And then within that white matter, there are areas of gray matter. And this includes the basal nuclei, which includes the caudate nucleus, the putamen, and the globus pallidus. We talked about some of this in lab. This is important for voluntary control of movement. We also have the basal forebrain nucleus, which is important for memory, the claustrum, which we don't know what it does, and the amygdaloid body, which is in the cerebrum, but is considered part of this limbic system that is important for integrating emotion and memory and things like that. And you see some examples of this here. Now, I'm glossing over this without getting into too much detail, and that's intentional because we could spend a whole semester talking about these structures. The takeaway message here is I want you to know that there are parts of gray matter deep to white matter in the cerebrum. And that's all I'm going to cover with the cerebrum. I'm going to spend the last part of class today talking about the protective layers of the brain, and that's going to wrap us up from our unit on the brain. Before I do, can I take any questions?
Will we have to know Wernicke's area for any tests or exams?
Oh, no, I didn't really talk about that one, so you can not worry about that one. I don't think we even mentioned it. I have to look back at the lab guide to see if I mentioned it in any detail in lab, but no, it's okay. All right, let's talk about how your brain is protected. This is a nice transition after seeing the video on Phineas Gage. By the way, I love the image in there, and they talk about the doctor seeing him and he vomits, and about half a teacup full of brain matter comes out of that wound. That's just an amazing image. Also, just curious how they approximated the volume of it in teacups, but whatever. Okay, the brain is protected from injury. So the brain is super important, but also super delicate and super sensitive. And it's delicate and sensitive to chemical imbalance. So it needs a constant supply of oxygen and fuel substrate like glucose, for example. But it's also very sensitive to physical injury. And of course, to protect the brain, we have a number of structures that help prevent physical injury to the brain. And this includes, of course, the skull, which you know very well, something called meninges, which we'll talk about here in a second. And that in between those layers of meninges or connective tissue, we find cerebral spinal fluid. And I'll also talk a little bit about the blood-brain barrier. So let's just put all of this into one diagram and take a look at it here. This diagram is key, and this shows you all of these kind of protective layers as they sit together. And I'm going to walk through them moving from superficial to deep. The first layer of protection is, of course, your skin. Here, the skin of your scalp, which Remember from early in the semester, contains the dermis, the epidermis, the dermis, and the hypodermis. All of those layers are present in the skin of your scalp. And just deep to that is the periosteum of the skull bone. And you can see the skull bone here. Remember that periosteum is that connective tissue that surrounds our bones. And of course, we have that layer of periosteum on both the superficial and the deep side of the skull bone. Now, the deep layer of periosteal tissue is along with this other layer of connective tissue that's just deep to that is known as the duramater. So this is an important vocabulary term you should know. The duramater being composed of this the deep periosteum of the skull bone combined with this meningeal layer of connective tissue. Now, these two layers are generally in contact with each other, and you can see that right here, that they're generally like flat up against each other, this periosteum and this meningeal layer, with the exception of a few places where we have something called a sinus, where those layers separate and they're filled with fluid. And I'll talk about the functional significance of that fluid in just a minute. Deep to this duramater layer, we have something called the arachnoid mater, or the arachnoid space. The arachnoid space is the, well, let me be more clear. The arachnoid mater is this layer of connective tissue just deep to the duramater. And then kind of deep to the arachnoid mater, we have something called the subarachnoid space. And it's called arachnoid, coming from the word arachnid for spiders, because it has this very spider webby appearance. And because of that spider webby spongy appearance, that's actually filled with fluid, specifically cerebral spinal fluid. Then deep to that subarachnoid space, on the other side of that cerebral spinal fluid, we have something called the pia mater. The pia mater, which literally translates as gentle mom, which is nice to think about, is continuous with the sulci and gyri of the cerebral cortex. You can see that here. So note that this came up, I forget who asked about this the other day in class, I mentioned it briefly. The duramater is kind of smooth and is more or less continuous with your skull, whereas this pia mater is continuous with all the sulci and gyri, all the grooves and bumps of the cerebral cortex. And so in between those two layers, we have the cerebral spinal fluid, which is kind of continuous with both layers surrounding the brain. Now, in this subarachnoid space, in addition to cerebral spinal fluid, we have blood vessels, arteries, and veins as well. And there's one other structure I want to bring up, and I'll show this in more detail here, but you can see that in this longitudinal fissure, so between the left and the right hemispheres of the cerebrum, we have the duramater kind of folds inward and actually goes between those two hemispheres and forms something called the falx cerebri. Now, this term probably sounds familiar to you because when we talked about the bones of the skull, I mentioned how the falx cerebri connects to that little bit of the ethmoid bone that sticks up called the crista galli. And you can see that here. So the falx cerebri is this kind of sickle or crescent shaped connective tissue that sits between the left and the right hemispheres. Notice that it doesn't completely divide them. In here, where we don't have the falx cerebri, this is where the corpus callosum is. So this is where those important tracts of white matter go that connect the left and the right hemispheres. This falx cerebri is attached to the crista galli, the ethmoid bone here, and basically separates the two hemispheres of the brain. Now, we also have a little bit of connective tissue here with the same structure called the falx cerebelli. And as you might guess, this divides the left and the right hemispheres of the cerebellum. And then we also have something called the tentorium cerebelli. This is, again, the same duramater tissue, but it covers the anterior portion of the cerebellum. So note these important terms here in blue. You should make sure you focus on these. And what's important to take away is that these structures are internal to the brain, but they're continuous with this duramater that surrounds the brain. I think that's the major points here. Yeah, that's all I'm going to say about that right now. Now, I got a little bit ahead of myself. I want to go back to this diagram. And I actually talked about all of these already, but I'll just review it. The arachnoid mater, remember, is this layer that's deep to the dura mater and creates this subarachnoid space. And what's important here is that there's these structures that I didn't talk about before, these called arachnoid villi, or here they're labeled arachnoid granulations, same thing. And what they do is they're actually little bits of this arachnoid mater that poke through the dura mater. And this allows cerebral spinal fluid to drain into these sinuses. And so the cerebral spinal fluid, at any given moment, you have a total of about, I don't know, half a cup of cerebral spinal fluid in between your brain and your spinal cord. And it's constantly flowing. It's constantly being processed through your blood vessels. And I'll show you how it's created in just a moment, but this is how that cerebral spinal fluid then gets dumped back into these sinuses, where it goes into your bloodstream and is circulated and essentially keeps cycling. And I mentioned finally here that pia mater is this layer of connective tissue that clings tightly to the surface of the brain and follows all those contours of the sulci and gyri. Now, the cerebral spinal fluid is this special fluid that is produced in the choroid plexuses of the ventricles of the brain. So we talked about the ventricles of the brain, the two lateral ventricles, the third ventricle, the fourth ventricle, and we had these beds of capillaries that are referred to as choroid plexuses. These plexuses are composed of ependymal cells and capillaries. So you remember ependymal cells, they're one of the 4 types of neuroglial cells. There are these epithelial cells, and the apical layer of these cells contains these cilia that move the cerebral spinal fluid around, and they help basically filter blood to create cerebral spinal fluid. Now, you create about 500 milliliters, so about half a liter of this every day from your blood. It cycles through, then gets dumped back in those sinuses and back into blood circulation. So at any time, you only have about 100 to 160 milliliters at any one time, which is not a lot. That's about half a cup, let's say. And so that means that half a cup of this fluid is inside your brain, surrounding your brain, and down that central canal of your spinal cord. So there's not a lot of it, but it is really important, and it provides a continuous layer to kind of move molecules around over the surface of the brain, but also to provide a kind of fluid protection from the brain to protect it from impact. And we can see what that looks like here. This is a sagittal section of the brain, and I'm not going to get into a ton of detail about the path by which this cerebral spinal fluid flows, but there's a couple important things that I do want you to take away from this. One is that the cerebral spinal fluid is produced at this choroid plexus, which is associated with each of the ventricles of the brain. So if we have 4 ventricles in the brain, we have 4 choroid plexi. Then that is in circulation, and that all of the cerebral spinal fluid is continuous, meaning that the ventricles of the brain, as well as the subarachnoid space, as well as the central canal of the spinal cord, all of those are connected, so that cerebral spinal fluid is continuously flowing through all of those areas. And then finally, the cerebral spinal fluid through these arachnoid villi or arachnoid granulations can pass through the dura mater and basically get dumped into these sinuses, where it travels, where it mixes with the blood in the veins and goes back into circulation. So again, the idea here is you're kind of constantly washing this cerebral spinal fluid across all of these different parts of the brain. Now, this layer, this protective layer, is important because it also prevents certain compounds in your blood from getting to the brain easily. So to get to the brain tissue, this blood has to pass through this choroid plexus and become this cerebral spinal fluid in many cases. And these capillaries in the choroid plexus are relatively impermeable, meaning that not a lot of things can get through there. And this serves to protect your brain from toxins or other molecules or things like that. But what's important to note is it's not an absolute barrier. While it does protect your brain from a lot of potentially detrimental chemicals, it does allow, of course, things like oxygen to pass through. Remember, oxygen is a small molecule, and it's also nonpolar, so it can pass through those layers of the capillaries pretty easily. Other molecules can pass through as well, including alcohol, which is why when you consume alcohol and it's absorbed through your digestive system, goes into your bloodstream and then can get to your brain. Nicotine as well, which can get absorbed through the lungs and get into your circulation of your blood, and then of course reach your brain. And things like anesthetics. That's why you can inhale an anesthetic before surgery, let's say. Again, that passes through the lungs into the bloodstream and then can pass through this blood-brain barrier. So only certain kinds of chemicals can get through this barrier, and not all. So it does serve as protection, but it's not an absolute protection. Ethan, you got a question? Go ahead.
Is this how infections of the brain occur too, like bacteria or viruses getting through that blood-brain barrier?
They can, yeah, that's a good question. And so specifically related to that, you've all heard of meningitis, which is an infection of the meninges of the brain. So the meninges being this layer of dura mater and pia mater. And that's exactly what happens is those layers can become infected. And that's bad news. And just in brief, it's really bad news because infections are also often accompanied by swelling. Now, the brain has a little bit of room to swell, but not a lot. And so what happens if you get swelling in these meningeal layers that are between your skull and your brain, you're basically, what ends up happening is your skull is not going to move much, if at all. And so that pressure then ends up getting put on your brain and can basically serve to compress your brain. And that's why infections of these areas are so dangerous, because while our skull serves very well to protect our brain from physical injury, it doesn't leave much room for swelling. And in many cases, they actually have to drill a hole through the skull in places to drain out some of that fluid so that that compression doesn't affect the brain. Good. I wanted to talk about that, so I'm glad I did. And that is it for the brain, people. That's all we're going to cover. We're going to transition, talk about a few other topics next week, and we're going to wrap up the course talking about the cardiovascular and respiratory systems. We'd be remiss if we didn't do that. And hopefully we'll have a little bit of time to touch on the digestive system as well. So I'm gonna just wrap things up here. I do have your Friday Venn diagram. I don't think I've used this one before. This one I thought was pretty clever. If you're from Nebraska, Arkansas, California, etc., you'll appreciate this one. Notice that Ohio is only on the U.S. state side of that. I don't know somebody had too much time on their hands to think about all that. Just a couple reminders that you've heard me say this many times, your lab practical will be during our designated final exam period, which I have to say has quickly come down the pipe. Semester's wrapping up, people. Also want to remind you that I will have open hours on Monday from noon to 2. Come into the lab room. We do not have a lab quiz next week, but you might want to come in and just kind of start studying for that lab practical now a little bit each week. Sure won't hurt. And then I want to remind you that quiz number 5 is on Wednesday, the 13th of April. There's details of this in your class program for Monday, which also includes a little bit more reading on these protective layers of the brain, and a couple videos I think will be helpful. The quiz is going to be in class. It'll be closed book, and it's going to only focus on the brain. So that is material we started on Wednesday, March 30th through what we covered today. And of course, let me know if you have any questions. So with that, I'm going to stop recording and wrap things up, and I will be happy to stay on and take any questions. Thanks a lot, everybody. Thanks again for being flexible, and I hope everyone has a great weekend.